First of all, what is science? Science is a way to explain the natural world. Everything that is happening around the natural world can be explained with the help of science. Science is a way of knowing and learning, which means science cannot be mixed with religion. Now, why do I say it? Because as a scientist, we can replicate the same experiment 1,000 times, but Jesus cannot replicate the same miracle 1,000 times. That is why we should never ever mix science with religion. Some aspects of religion can be explained by science, but we should keep them apart every time. Now, science is based on evidence, and science is tentative. What do I mean when I say science is tentative? It means that it is subject to change, and it changes because of new technology that is coming as we go forward. So science is tentative because of the new technology that we have each day and is based on evidence. Very good example of science changing with technology is when we define solar system. Earlier, it was supposed that Earth is at the center of the solar system, but after we had the right technology, we could say Sun is at the center of the solar system and not the Earth, and all the planets revolve around the Sun. The evidence that Nicholas Copernicus based his theory on that Earth is at the center of the solar system, it was based on statistics and maths. Whereas Galileo, he had telescope, he had technology, and he could provide evidence that not Earth but Sun is at the center of the solar system. So, what is the scientific method? It is based on making an observation and then asking the right kind of questions. In science, we need a method so that we can explain the natural phenomenon that is happening around us. First of all, we make an observation. For example, I made an observation that male Madagascar hissing cockroaches look larger than the females. Now, then I ask myself a question that why is that? Why males look larger than females? And is this a thing in Madagascar hissing cockroaches? Then I will form hypothesis. Now, hypothesis is based on three things: making an observation, providing an explanation, and then giving a prediction that if you perform an experiment, this will be your result. So I made an observation that male Madagascar hissing cockroaches look bigger than female Madagascar hissing cockroaches. Now, what can be my explanation? For example, males fight, right? They have to show aggression. And if big size is an advantage, then males should be bigger than females. Notice that I have an observation that males are bigger. I provided an explanation that bigger size is an advantage when they show aggression. Now, the last thing left is prediction. I'll say when I do an experiment in which I measure the length of different Madagascar hissing cockroaches, uh, ultimately the males will be bigger than the females. Now, this is a prediction, not a result. So, we predict our experiment results beforehand so that we can reject it or accept it. Now, we perform our experiment and then analyze the results. If we have conclusive results, we report our findings that yes, male Madagascar hissing cockroaches are bigger than females. I have conclusive results. Then we report our findings. But if we don't have any conclusive results, we can always go back to changing the hypothesis. We can change our hypothesis or change the way we performed a particular experiment. We don't interfere with the results. We don't falsify the results. We change the hypothesis. It is not a good solution. It is a very bad solution that we interfere with the results. We can always change the hypothesis. So Alexander Fleming, he discovered penicillin, and he discovered it by chance. He had a very he did not keep a very neat and tidy lab, and one day he observed that one of the petri dishes don't have any bacterial growth at a particular spot. Now, then he made an observation that yes, these bacteria are not growing in this particular spot. Then he asked a question, why is this bacteria not able to grow whereas all other bacteria are growing? Then he made a hypothesis that something is going on that is hindering the growth of bacteria, and then he made replicates, and while he took the sample from that plate, and that is how he was able to discover penicillin, because he made the right kind of observation, he asked the right kind of questions. That is why we have penicillin, which is a good antibiotic. The next thing is peer review. What is peer review? Uh, when we are done with the experiment, we are done with the results and conclusion and everything. The author submits an article. Then this article is assessed by the editor of the journal, and then the journal sends the article to the reviewers, which review the article. Then these reviewers decide that this article is okay to be published, or it needs revisions, or it is rejected. So the editors and the reviewers, they review the articles and come up to this conclusion that okay, if it is accepted, it will be produced and then published. But if it's not, then they will mention the reason behind it. That the reasons might include that your ideas are not unique, this research has been done before, or your methods are not correct, or you need certain revisions in particular section of the paper. It can be anything, and the author then will consider these revisions and then again submit the article. And this is called peer review. Now, talking about the experimental design I discussed in the previous video that was uploaded on Blackboard earlier. It had an experiment in which you use two kinds of water, that is rainwater and normal water holes, to water rose bushes. Now, in this particular experimental design, our experimental variable is the kind of water we are using. We only need to have one experimental variable, and that is our kind of water. The next thing, we're going to make sure that everything else is controlled so that we have a good experiment. Now, while changing only the kind of water used to water the plants, what else do we need to control? Suppose the type of rose bush. If we are using pink roses, then we are using pink roses and not red roses. The kind of fertilizer we are giving both the plants, the amount of sunlight they are receiving, the amount of water, and the temperature that both the plants the temperature that both the plants are kept in. This needs to be controlled. So only one experimental variable and all other variables should be controlled in order to have a good experimental design. And when you replicate your experiment, you should consider all these things and then perform replications in order to be sure that your results are conclusive, and then you can submit your research. So in this lab, we were supposed to design a class experiment using Madagascar hissing cockroaches, and we were supposed to measure the length of different Madagascar hissing cockroaches, that is males and females. Now we can only suppose that we did it, and we will go forward from there. Before going over the experiment, some things that I need to remind you of is documenting in your lab notebook. You should always do it with ink, don't use pencil, and record as you work. Don't leave any blank spaces. If you have a blank space in your lab notebook after completing that lab portion, just cover that blank space with a diagonal line and put date and signature in your lab notebooks. Then don't remove any pages from a lab notebook. So next is our experiment that we're going to do in our class. Change the date. The date is whenever you do the experiment. That is 17th of February we were supposed to do it. Objective to determine if adult male Madagascar cockroaches are longer than adult female Madagascar cockroaches. Hypothesis: if male MHCs rely on aggression to compete for resources and bigger size is an advantage, then the average adult male MHC will be longer than average adult female. Now for the procedure. Now in lab 1 we used just a ruler to find out the length of the different Madagascar cockroaches. But MHCs are not straight. They have curved surfaces. That's why we would use twines or threads in order to measure the length. We will put one end of the twine on the head of the cockroach and measure it until the tail, and then measure that twine with the help of a ruler. That will be the exact length of the Madagascar cockroach. Now suppose we have we have measured the cockroaches, and in each lab we have 2 males and 2 females, and we have these measurements: 50 millimeters, 60 millimeters for males, and 65 and 60 for females. Don't forget to put your signature, and you can copy this as such in your lab notebooks. Next is documentation of observations. As I said, if you have any blank spaces, this is how you're supposed to cover them. Draw a diagonal line on that blank space and put date over that line. And again, don't forget to signature at the bottom of a lab notebook. Now we have the data. From the data we could see females are slightly bigger than males, but can we just conclude our results on the basis of that? No, that doesn't make any sense. We need to do some statistical analysis in order to actually say that yes, the males are bigger than the females, or the females are bigger than the males, or there is some difference in the size, but we don't know yet which one is bigger than is there any sexual dimorphism in size. Now how can we do the statistical analysis? For that we will use t-test. Now t-test measures the difference from the mean of each observation according to the variance from the mean. If you see the numerator, it compares both the means, that is x and y. X is the mean average length of females and y is the mean average length of males. Whereas in the denominator we have standard deviation, that is Sx for females and Sy for males, and n represents the sample size. So the denominator accounts for variability and sample size. And if you have a larger sample size, then your outliers will have less effect on your data. So what do you mean by outliers? Suppose if I have a class of 100 students and the average score of the class is around 85, but there are some students that have a score of 35 or 40. Now these are my outliers, but I have a class of 100 students. Having a large sample size does not affect my analysis, even if there are a couple of students with such low scores. So if the sample size is large, your t-test, your statistical analysis will not be affected as such. In this case, you have only two replications, that is of males and females, 2 males and 2 females, so your sample size is really small. Now T is directly proportional to the difference in between the between the averages of males and females, but it is inversely proportional to the standard deviation, to the variance. So increasing the difference between the averages will yield into a larger t value, and having less variance from the mean will give us a larger t value, whereas having more sample size will also give us a larger t value. So in order to have a larger t value, you should have 3 things: a bigger difference in between the two variables, averages of those variables, less variation from the mean, and a larger sample size. Now t-tests are of two kinds, one-tailed and two-tailed. When I'm saying one tail, what I mean is there is some directionality in which I'm going. For example, when I say males are supposed or males are bigger than the females, that I'm going in one direction. I'm not saying that okay, either the males are bigger or the females are bigger. I'm going in one direction. That's called directionality. When I say something is bigger or smaller, when I say something is more or less than the other, or higher or lower. So when you go in one direction, when you go in one direction, that's a one-tailed t-test. And what you did uh, was a one-tailed t-test. Now, when you are going in two directions, when you're willing to go in both the directions, bigger or smaller, that is, we don't know that which one is bigger or smaller, we just know that there is some difference. We are willing to go in both the directions, then it's a two-tailed t-test. All right, and in a two-tailed t-test, we have a more stringent test. The test becomes more, uh, how do I say it, accurate, and we will have more accurate values of p. When I say t-test, you don't have to do it on your own. There is an online calculator that you can use. This calculator looks somewhat like this, in which you put the treatments for X and Y. The two male were that were taken, that is one was of 55 millimeters and the other was of 60 millimeters, and the two females, one that were taken, the one female was of 65 millimeters and the other was of 60 millimeters. Now that you have put the values, you need to set the significance level. When I say we need a significance level of 0.05, what I mean is I need my results to be 95% accurate. I'm only leaving things to random chance for up to 5%. There are three different levels that you can choose, but in our analysis we will always go for 0.05. We only leave things up to 5% on random chance, and we need 95% accurate results. Now, when I said one-tailed or two-tailed hypothesis, remember that our hypothesis was males are bigger than the females. We were going in one direction. That is why this test becomes one-tailed t-test. Now, after selecting these options, you just have to hit on calculate t and p values, and it will give you some results. This calculator will take you to another window in which it will, at the bottom of which it will tell you the values of t and p. For this particular test and for these values, we had a t value of minus 1.41. Remember one thing, t value is never negative. The calculator cannot differentiate that it is an absolute value, so t value is always positive. So if it says minus 1.41, you're not going to consider the minus sign. T value is always positive. The p value is 0.14, which is way over than 0.05. So our results are not significant, because in order to have significant results, we need a p value which is less than 0.05. So before, our hypothesis was that adult male MHCs are longer than adult female MHCs because they rely on aggression and bigger size is an advantage. But we did not get any significant results. That is why we are now changing our hypothesis. Now our hypothesis is that if MHCs display multiple forms of sexual dimorphism, then male and female MHCs will be different in lengths. Now notice that I'm just saying that they should be different in lengths because there is some sexual dimorphism. I'm not saying whether the males are bigger or the females are bigger. I'm willing to go in both the directions. So this becomes a two-tailed t-test. Previously it was a one-tailed t-test because I had direction. I was saying that males are bigger than females, but we did not get any significant results out of that. Now we don't interfere with the results, we change our hypothesis. So that is why my hypothesis becomes that yes, there is some sexual dimorphism, there is some difference, but I don't know whether the males are bigger or the females are bigger. Now if you perform a t-test using the two-tailed option on the calculator, you will get a t-value of 1.41, which will remain the same. The t-value does not change, but the p-value increases further. Now it says that 29%, that is 29.2% of my results are dependent upon random chance. and only 70% of my data is correct. Uh, this proves that when we do a two-tailed t-test, the test becomes more stringent and the p-value increases further. So in conclusion, we can say that our data does not support the hypothesis, and the results suggest that there is no significant difference between the length of male MSCs compared to female MSCs. But if we increase the sample size, if we take like 50 males and 50 females, we can have conclusive results. So whenever you're performing an experiment and you don't have significant results, it doesn't mean that your hypothesis was wrong. First of all, check whether you did a good number of replications or not. Replications is the key. You need to have at least 10 to 15 replications in your own long-term experiment. If you have only 5 to 6 replications, your t-test will not be as significant as it can be. Now that we are done with our experiment, this is about your homework now. So there are two types of scientific literature. First is primary literature. Now the kind of research that you will do in your class, building a hypothesis, and then writing all the procedures and then concluding your results. If you publish that research, it is a primary research. So it is basically an investigation that is conducted by the author. Whereas secondary papers, they summarize all the primary literature that has already been published. Suppose I want to study cheetahs, but I don't have any cheetahs that I can experiment on. Therefore, I will study all the papers that have been already published on cheetahs, and then I will draw my conclusion, and then I will say that okay, I have read this and this paper. I think about this and there is a research gap that needs to be filled, and here is my conclusion. And when you summarize all the primary literature on a particular topic that you are studying, this becomes secondary literature, where you do not research on your own, but you already review the research that has been published. Now, what about your homework? Your homework is to find out 3 articles that have been published on the topic that I have given you. They should be peer reviewed, and it means that all the journal articles are basically peer reviewed. You don't have to look for any specific articles. So you will find 3 articles on your topic that I have given you, such as Madagascar hissing cockroach and reproduction or aggression or life cycle, anything. So you have to cite them using this particular method of citation, this particular protocol. So the authors come first, then the date, then article title, followed by journal title, and then volume. I know this is very similar to APA style, but APA style has also has links. I don't want the links, so remove them. And along with the citation, you need to provide me with a summary of the article. Now it can be a 4 to 5 line summary. It can be a 6 to 7 line summary. Just don't provide me with one-liners. You can find articles on googlescholar.com, or you can also find articles on TTU website, because all the articles that are on Google Scholar are basically peer reviewed. So what you will do is you will write your name, the kind of article it is it a primary article or a secondary article? Then you will cite it, provide a summary, and then provide me the screenshot of the PDF file of the article, first page of the PDF file. Don't take screenshots of websites, just the PDF files. If you still have any confusions, there is an example on Blackboard. You can look at that and just copy that example 3 times. All right, so follow that particular criteria of homework in order to get full marks. It's a very easy one. You can do it. So you need to design your own experiment, and for that you will have 4 weeks. Starting off each week, you will have 10 minutes. So if your lab starts at 2 o'clock or 10 o'clock, you will have until 10:10 or 2:10, and then we will proceed with the particular chapter You will only take quantitative data. You will not take qualitative data, as we have discussed earlier. In all the scientific investigations, quantitative data gives us results and not qualitative one. You will only have two treatments, not more than two treatments, and only one experimental variable. For example, if your experimental variable is food, then you should only change the food and not anything else. And if your experimental variable is like the sex of Madagascar hissing cockroach, then everything else should be constant. So all other variables should be controlled. Replications as much as possible. Do as many as you can. So basically go for like 4 to 5 replications each week. And you need to get my approval after selecting the topic. So we have these 6 research questions that you can choose from. The first one being, are MHCs motivated more by fresh or dry food? So in this one, you will have two different kinds of food. The dry food, we usually use the mixture of dry cat and dog food. For fresh one, you will have a piece of carrot, or you can bring food from your own, like a strawberry or something. But you need to keep that constant. Don't change the type of fresh food. So you will take a maize and keep for one replication you will take fresh food and the other one will take dry food. You will see when how long the MSC takes to finish the maize in presence of both of them, like fresh or dry. The next one being, are male or female MSCs faster? Now you will take one male and one female MSCs and then time them and see how long they take to complete the track. The next one being, are flipped MSCs able to write faster on a smooth or a rough surface? So you will take MSCs and flip them first on a rough surface and then time how long do they take to write themselves up on their legs, and then on a smooth surface, and you will compare the difference between the two. The next one being, do MSCs move faster in dark or lit conditions? They will be the same track, just you will cover the track with your lab manual or your lab notebook for the dark conditions and just keep it open for the lit conditions. Now, do sounds influence the ability of MHCs to complete a maze? Same track. First, MHC will complete the track without the sounds, and then the same MHC will complete the track with the sounds, and you will compare the difference between the two. The sounds can be from YouTube, like jungle sounds. Don't play a song, just like nature sounds. Are male MHCs more prone to right or left antenna breakage? Now, we all know that male MHCs fight, they show aggression, and in that aggression, in those fights, they tend to lose or they tend to break their antenna. So what you will do is you will only collect male MHCs and measure the length of the antenna with the help of a ruler and see whether the right antenna is bigger or the left one. So this will be a comparison. Now, after choosing a topic, you have to fill your second page of lab 3 in your lab notebooks. Write your objective, your hypothesis, and your procedure that you will follow to do the experiment. Before doing that, you will have 72 hours to email me about your topic as a group, and I'll let you know that is this a good option, is this feasible, and please get it approved, and then go for the next homework. The next homework is going to be experimental design worksheet. An example of this is already uploaded on Blackboard, and there's one file that is fillable. You can download it, fill it, and then email it to me. You will have a week for this assignment. So 72 hours for choosing a topic and a week for giving me the long-term experiment worksheet. The worksheet includes questions such as state your group's research question. Simple. So I'm going to explain it according to the like this lab. Are male Madagascar hissing cockroaches longer than female of the same species? What are some explanations for this observation? So explanation is males are aggressive and females are not. Develop a hypothesis about your research question. State your explanation. Again, male MSCs rely on aggression and bigger size is an advantage. That can be an explanation. State run prediction. The average adult MHC will be longer than average adult female. Very simple, right? Similarly, we have think of an experiment you could perform to test your prediction. So the experiment is uh, measuring both male and female cockroaches, in which our treatments are adult males and adult females. Experimental variable is the difference between the two treatments, that is the group, that is the sex. How many cockroaches with your use for each group? So I gave you an example of using only two cockroaches, two males and two females. Why did you choose a number of cockroaches? Because uh, so in this particular case, how many cockroaches with your use for each group? You can say that at least 14 to 15 for your long-term experiment. And why did you choose this number? You can say that you had 10 minutes like in the beginning of each lab, and in those 10 minutes you could perform 4 to 5 replications each week. So 4 weeks, maybe 15, maybe 20 replications, as many as you can. What variable will you measure? So in this particular experiment, where we measure the lengths, how will you measure it? We used Twines. Explain the variables you will keep constant. So this is very important. You need to keep the species that we are using, the Madagascar casing cockroach. You are not using any other cockroach. You are using this adults and not the juveniles or the babies. You're using the same measuring equipment for all the cockroaches. If you're using twines and rulers, you're using twines and rulers for everyone. Should you repeat your experiment? Why or why not? That is dependent on your long-term experiment. If you have significant results, you don't need to. But in this class, we did not had any, so that's why we need to. What are the independent and dependent variables? Now, you need to understand this carefully. So the independent variable in this experiment was the sex of the cockroach. Therefore, the dependent variable becomes the length. Why? Because dependent variable, that is the length, was dependent on sex. If males are bigger, then the length will be bigger. So dependent variable always goes on the y-axis, and the independent variable always on the x-axis. Make sure of that. And you will only make bar graphs. No line plots, no scatter plots, just bar graphs. Describe what conclusion you could make if your results supported your research, your hypothesis, sorry. And you can state that yes, male Madagascar hissing cockroaches rely on oppression, and that's why they have a bigger size. If not, then you can say that there is some difference, there is some sexual dimorphism, but so aggressive behavior has no effect on size within the population. So in the end, you have to take care of three things. Your preliminary exercise, you can submit it by Friday, and I'll provide you the exact timings and the deadlines for that in my email. You'll have to choose a topic and let me know, and fill out the lab 3 according to your research topic that you're going to choose. Please choose out of the 6 topics that are provided in this video. The last thing is the long-term experiment worksheet. So this worksheet is of 20 points. Be very careful while filling it out. Discuss it with your lab partners. Have a virtual Zoom meeting or meet face to face. Just the deadline is the deadline of that worksheet will be Thursday 11 p.m. So that's it. If you have any confusions, I'm gonna have my Zoom hours on Monday, and you can get your topics approved on Monday as well. And if you have any questions, just we can discuss them on Monday.